Hi, Chris. Thank you for joining this conversation.
Sure, it's great to be here. I'm excited.
I'm glad you can share some of what you're working on and some of what got you to where you are now. So let's start with who you are and what you're working on. Do you want to introduce yourself?
Yeah, absolutely. So I'm Chris Kuffner. I'm 25 and I'm a third year PhD student at Boston University in the biomedical engineering program. Right now, my research is heavily focused on protein engineering, which I use some synthetic biology tools to do. And I'm particularly focused on protein degradation and its application for imaging single molecules within cells.
That is fascinating. I was just talking with somebody from an industry partner who focuses on protein degradation. I think it's a tool for observing and engineering cells and understanding cells that tends to get overlooked. So is your lab exclusively focused on protein degradation, or the lab that you are getting your PhD in, or are there, is it part of a bigger whole question that they're asking?
Mhm. My lab is definitely focused on protein engineering. So some labs will take a more bird's eye systems level approach. They won't get into the nitty gritty of single proteins, but we really dive into the protein itself. Uh, Although I am sort of the resident degradation person, and everybody else does different variations on things that proteins do.
Well, I always say proteins are doing the bulk of the work of the cell, so you have lots to choose from. So protein degradation, that is something that's difficult to observe for sure. You must be using very interesting tools to be able to understand a little bit about how proteins degrade and when they degrade. Do you want to talk a little bit about like what your work is focused on and doing?
Sure, I won't get too into the nitty gritty, but throughout my career, I've always been interested in tools and specifically making them easier and more accessible. So when I first was starting on the protein degradation project that is now the focus of my PhD, I was having a really hard time working with some of the tools that are existing. And more than anything, I really want to make them more understandable, easier to work with. So I don't think I'm ready to share the details, but I believe that my systems are a little more flexible, a little more understandable than the others out there.
Yeah.
And yeah, that's important to me.
That's very cool. You know, frustration with how things are working often leads to great engineering breakthroughs. I think the washing machine was invented for that reason, and all kinds of things. So yeah, I think the appreciation that things should be and could be done better definitely leads to innovation and advancement. So how is it going, just sort of generally? What's up? What's exciting?
Well, I have some exciting videos here, so hopefully these show up nice on the recording. But Natalie, just checking, can you see this?
I sure can. Wow, that is beautiful.
It's really exciting. So I've used protein degradation to improve methods for single molecule RNA imaging. Right now, you're looking at single RNAs in a cell and how they move throughout the cell. And we can use this system to observe how RNA moves depending on what the cell is making with that RNA. So this is an RNA that is very free to move within the cell, but then in some other cells like this one that are expressing a different kind of RNA, it's much more static. It moves less within the cell. So that's just a little preview of what I'm working on. But I think that they are very cool images. I'm very proud of them.
That is amazing, amazing. And you know, it's like the inner life of a cell, right? Like it's easy to forget there is so much going on, that there's like differences in rates, differences in concentrations, and that can really be indicative of what the cell is up to. So how amazing. That's awesome. You must have been so excited when you saw those images.
Oh yeah, really,
Yeah.
really exciting.
Yeah. So making cool tools, finding great data, it makes it sound like the life of a PhD student is pretty awesome. are you, do you have ideas about, are things about your PhD as you imagined they would be, or different than you imagined they would be?
Sure. Well, it goes without saying that the pandemic put a wrench in everyone's spokes, but right about now, things are going well. I think when you've been a scientist as long as I have, you acquire an understanding about how some things work and some things don't. And it's important to be aware of this and know what direction will be fruitful and which ones will lead to a dead end.
Yeah.
I know that outside of academia and industry, they take that to an even greater extreme. I've heard that, if they don't get results soon, it gets dropped very fast. I think one of the greatest things about the PhD that you really won't find in any other scientific opportunity is, several years to focus on one specific project. I'm not going to lie, it took a while to get to the point where I can make those nice images. And I think that thanks to the PhD, I was able to dedicate the amount of time towards doing that, even though this isn't, for example, something that I can just immediately sell to people. It's just, this is how scientific discovery is made.
It is. And I will say, I think it is the investment, especially here in the United States, that we make in our PhD students and in our fundamental research that has enabled our country to stay ahead and advanced in this entire field. So you're right. It is a rare thing to be able to take the time to look at questions that, in early days do not seem like they're going anywhere, but to be persistent with them and to be dedicated to them as a student to make them work and to really apply your best. So yeah, it is how we maintain our success and our future look at biotechnologies that you're doing. So that's awesome. So let's see. So it sounds like, at least, especially at this moment, your PhD is going pretty well. Where had you come from prior to starting your PhD? What did you study that led you to this step?
Sure. So thanks to some early experiences in BioBuilder, I've always been way into synthetic biology and bioengineering. I've been especially obsessed with really getting inside the cell and reducing things down to that, DNA, ATCG equivalent of ones and zeros on a computer, where you feel like you just have so much precise understanding and control. And honestly, that's why I really like my project now, is because of that single molecule imaging. It's like way back in the olden days when the first microscopes were coming out, and you simply just looked and saw what hadn't been seen before. So, oh gosh, where was I going?
Well, I mean, I think it's fun. The notion that we can make the invisible visible is so powerful and so exciting, right? And when you're saying looking through the microscope, right? Like, think about Hooke looking at that cork under the microscope for the first time and seeing what he saw and knew were cells and identified what he called as cells. So being able to witness properties of the cell, behaviors of the cell, activities of the cell has been just an incredibly revealing tool for biology and applications that you wouldn't necessarily anticipate just by knowing how things look. So very cool.
Yeah, and I just remembered the question. When you're a PhD student, you get so into your project that it's all you can talk about. I love this project, but I didn't really clarify. So it started with BioBuilder and that passion to really get into the cell, into the molecule, and get to those details. I will say that the beginning of the career was, and sometimes it was frustrating because it's not easy to do these things. I remember way back in high school, I found out the hard way that lab equipment is pricey and high schools don't have that equipment. So, throughout college, I was actually doing projects where I tried to sort of build the equipment that I was missing in high school from little parts and scraps. And that actually worked sometimes. I was able to make, for example, a freezer that would normally be a very expensive lab freezer with parts for cheap. And another thing that I was able to do was make a biosensor that could detect an interesting phenomenon, which would normally require extraordinarily expensive instruments to look at. So, yeah, I think right now I'm at the real science stage, but before now, I think I was really just focused on how can I make real science possible? Things like this. That's where I was before I was a PhD student.
Yeah.
How do we get here?
Yeah, I mean, democratizing the tools, democratizing the information, making sure that anybody who is curious has access to it, is so aligned with the work that BioBuilder does and has always been sort of founded on. I think that that's it makes perfect sense that we would have connected early in your career. So where were you in high school? Where'd you go?
I was in Massachusetts, Acton Boxborough High School, and shout out to my teacher, Aaron Matthew, who currently runs BioBuilder Club there. Just really excellent teacher. The one thing I remember most about my experience in high school was the ODAT smell lab with the banana scent E. coli. I just remember being astonished at how a little bit of synthetic biology could make this bacteria that smells like a landfill smell really, really tasty. That was kind of the moment where it all clicked to me that, wow, this is incredible. This is going to change the world. And I was committed.
Wow. Well, it's certainly gratifying, as part of the team that brings that out to the world to hear that it has influenced you and helped. And yes, a real shout out to Aaron at that high school. He's amazing. So awesome. And then you went to college and studied bioengineering. Is that right?
Yeah, I studied bioengineering, and this was at the University of Maryland. They had a really great curriculum, and I really liked the environment there. Yeah. I also was involved in iGEM during college, which I think can be a really great experience. Personally, I dig BioBuilder's approach to synthetic biology, but iGEM makes a great university approach where people can really come up with their own ideas and pursue these projects. This is actually where a lot of my research before my PhD came from.
Yeah, amazing. That's so awesome. So let's see, if there were a high school student listening to this conversation, is there any particular advice or lessons that you would have, things you would tell yourself, your high school self, things you've learned now that you might advise or have sort of greater attention to?
This synthetic biology endeavor is huge. It's bigger than me, it's bigger than you. Everyone has something to contribute. And sometimes the dream is to, for example, be like maybe a PhD with all my fancy instruments and my 5-year projects, unlimited time, unlimited money, let's do all the hard stuff. But sometimes you're in a position where you need to consider what is possible. I think if I were a high schooler, I'd like to tell myself, you may not be able to do some of these cutting edge things that you see in the newspaper, but you can, for example, totally learn a little bit of coding or automation, and you could make some cool devices that would help people work better in their labs. That's actually something that I'm personally very interested in for the future of our field. You can also read more about policy in synthetic biology, this brand new technology, and we don't know exactly what it could lead to. This is something that you could read about. The one other thing is maybe learn, if you're very interested in research, about the academic publishing process. I think BioBuilder does a really great job of teaching people this through the Biotreks journal, but I sometimes wish I'd known a little more when I was an undergrad, because I was approached by publishers once or twice and I actually turned them down because I didn't understand just how significant a publication is for anyone interested in a PhD.
Yeah.
I did okay, but
yeah,
sometimes
publishing is certainly a currency that that matters in this field, and that is a way of communicating the accomplishments that you've had. And thank you for the shout out. I do think Biotreks is a great journal for high school synthetic biologists, gives them a good taste of what peer review looks like and how to help people make the best journal article that there is, to have an outside pair of eyes on it and respond to comments and feedback. So yeah, I think you've definitely touched on the very broad road that it's a wide on-ramp to a career in synthetic biology, right? There are lots of things that this field needs, whether it's policy or publishing or hardware, and obviously the wetware and things like that. There's a lot of place for people in this field. And I think, engaging the next generation as the problem solvers and as the future leaders in this, we can do that real early. And I'm excited that mattered to you, because clearly you are leading the way and have developed incredible tools that are going to help a lot of people. So congratulations. It's exciting. I'm excited to see what happens next.
Thank you.
Yeah. And thank you for taking the time to talk to folks today and share all that you've done, your wisdom, your experiences. It's been great.
Mm-hmm. And, just to anyone who's listening, good luck. This is such an exciting field. I think that, thanks to what a lot of people out there are trying to do, you're going to have so many cool things that I didn't have when I was trying to get into the field. And the only thing that I really ask of you is that you keep trying to make things more accessible. Please pay it forward. Think about mentorship and maybe tools to help other people who want to be what you want to be.
Yeah. Pay it forward. Absolutely. Definitely. Thank you.
Thank you.